Hi, Teresa. So welcome back, everyone, to this afternoon session on analytical approaches. I've been very excited about this session. To start us off is going to be Teresa Shatika from the NIH's National Center for Biotechnology Information. And the title of her talk is Mutational Signatures as Tools to Study Interactions Between Environment, Cellular Processes, and Diseases. Maybe while Teresa brings up her slides, I'll just mention that be sure that you put your questions into the Q&A, and I'll try to get to as many of them as time permits.
Okay, I hope the slides show up okay.
Yes.
Okay, so I would like to start by thanking for this kind invitation to present our research on this wonderful workshop. My group is a computational group, and we have been working actually on challenges related to mutational signatures for quite some time now. And today I'm going to discuss some of our most recent results. And our most recent focus was on interaction of mutational signatures. We are actually interested in two types of interactions. Let's get rid of namely interaction between signatures and molecular pathways that we study by combining mutational data with expression data. And obviously, we know that for many mutational signatures, the corresponding mutagenic processes might be unknown, and even if they're known, are not always fully understood. So by understanding how those processes interact with molecular pathways may provide a clue for related to their function. In the second part of my talk, I'm going to switch to different type of interaction, namely interaction between mutational processes themselves. So as you've seen from the Ludmil introduction and many other speakers, we tend to model mutational processes as being additive. That is, that we assume that starting with the mutational catalogs of all these patients, we can represent those mutations as a linear combination of mutational signatures. As gets mentioned, that's not always the case, that this additive assumption is not always correct one. So in the second part, I'm going to show some of the models models that we developed to deal with this challenge. And this will be modeled to model interaction between DNA damage and repair deficiency. Okay, moving to the first type of interactions, the studies that I want to describe today was motivated by the question whether or not smoking has positive or negative impact of progression of COVID-19. So in initial results related to this question were really mixed. So we thought that perhaps we can use mutational signatures as a window to study the relation of smoking with cellular processes. Now, the one issue is here that we really would like to understand the relation of smoking in cellular context that is not necessarily a cancer context, while the mutational signatures are usually available for us mostly for cancer data. So in this study, we selected lung adenocarcinoma since it contains quite a few samples that are control samples. And we analyzed the interaction between mutational signatures, which we are reading out from bulk cancer sequencing, and the expression of those in those control samples. So this is a caveat of the study. In order to be able to interpret this, we have to have a quick look at what are the mutational signatures that are known to be active in LUAD. So first of all, there's the smoking signature, signature 4. And for this signature, it's really, we treat them as a readout of smoking exposure. And it really doesn't matter that we take the readout from cancer and apply it to control tissue. The second pair of signatures, signature 2 and 13, are ApoBec signatures. Those signatures are often correlated with various inflammatory response, and we know that smoking often creates such response, but there might be some other cancer-related cancer-related causes. So this one we have to be open-minded for how we interpret them. Next to signatures 4 and 5. Those are both not well understood signatures, but are both clock-like signatures. And from our previous study, we have reason to assume that signature 5 is an environmental signature. Finally, signature 1 is definitely not environmental. This is a signature that is related to replication rate. So it will be definitely different in cancer in control. So we start introduction. Perhaps the first question before we move to looking at how we can utilize this interaction between signature and molecular pathways. Well, let us answer the simpler question. There is a number of proteins that are known to or postulate to facilitate viral entrance to the host cells. So what is the correlation with those viral entrance proteins? And we can see that those different bars corresponding to different viral entrance protein, and the correlation of most of them with smoking or those other environmental signatures is positive. So with that, now we would like to have something more robust and unbiased. And to perform this type of analysis, we developed a simple actual method, but we found it quite powerful. We refer to this ECO-seq class. So basically, the idea is that we start from the assumption that we have expression data and mutation data from the same patient. And this allows us to compute for every signature and every gene a correlation between signature, the mutation count assigned to the signature and gene expression. This allows us to build this gene by signature matrix, and we keep here only the genes that are significantly correlated with at least one signature. Then we take this matrix and cluster it so that the genes that have the same relationship, the same correlations with all the signatures are clustered together. And once we have those clusters, we analyze, and actually even simple Go enrichment analysis is quite powerful in this setting. So this is what the heat map of such clustering obtained for Lua data. And again, you remember that in this case, we are looking at control samples, and you can immediately see that there are two clusters that are positively correlated with signature 4. One is this cluster 5, which actually contain ApoBec gene, and its Go enriched annotation shows cytokine-mediated signaling pathway. And this is very, reassuring, because we really would expect that smoking would be activating cytokine-mediated pathways. The second positively correlated cluster is also immune response, also associated with innate immune response. So what is the relation, if any, to COVID-19? So we overlap the genes in both of those clusters with genes that have been identified as significantly upregulated in COVID-19. And those genes were, the overlap, I'm not showing here the p-value, but was really striking. There's also some other patterns here that are actually very interesting. Sorry about it. In this type of analysis, it's important to keep in mind that lung is a complex tissue. And what may be going on also is that in response to smoking, we have a change, we may have changes in cell type composition. A couple of previous papers suggested that such processes might occur. So we can actually capture those processes using mutation signatures. To do that, we modify slightly our approach, and rather than looking at gene expression, we use a program called CyboSort that allows to identify cell type composition based on bulk expression. It's not possible to do it for any tissue yet, but for lung, we have enough information to perform this type of composition. So again, we do now a correlation between strength of the mutational signatures and cell type proportion. There is no need now to cluster anything. Each row corresponds to one cell type, and what we obtain is extremely striking. So we analyze epithelial cells types and immune cell types separately so that we can see how proportion in each group is changing without considering whether immune cell increase overall. I will not have time to go through all the interesting stuff that this heat maps reveal, but for example, we can see that signature 4 increases the population of goblet cells. Goblet cells are mucus producing cells, but at the same time, signature 5, which is known to co-occur with signature 4, is decreasing, is associated with decrease of ciliated cells. So what's happening in smoking patients is that mucus producing cells are being increased, but mucus removing cells are being decreased. So that's certainly no good. There's other also interesting changes in the proportion, the reduction in CD8 plus memory effector T cells, etc., which clearly indicates that smoking predisposes individual to rather unfavorable disease progression. Now, I would like now to switch the gears, and I would like to move and discuss the other type of interaction, the interaction between mutational processes. So as been mentioning several times already in this conference, a typical way in which we use to infer mutational processes is by decomposing the mutation matrix into linear combination of mutational signatures. So this is illustrated here on this example that basically we assume, ignore for a moment this green region, but basically we assume that we have a number of mutagenic processes. Each of them is created, hits the genome with certain probability that is defined by the signature. And then those all mutations accumulate. But if we haven't seen this process and would like to understand what is the contribution of all these individual mutagenic processes, we run, for example, non-negative matrix factorization, but other approaches also use this assumption that the mutations that we observe as a linear combination of additive processes. This is not always true. I'm going to call those processes for which this is true, like smoking, UV light, and others, primary mutagenic processes. And why this name is because we have also what I'm going to call secondary mutagenic processes. And as a kind of a key example of this secondary mutagenic process is deficient DNA repair, for example, mismatch repair. Mismatch repair, deficient mismatch repair is really not so much introducing new mutation, but rather is failing to repair mutation that our process introduce. So if cell has deficient mismatch repair machinery, the type of mutation that we are going to observe at the end of the day depends on what primary mutational processes were active. So, in summary, we have two types of processes. One we call additive, which I'm going to refer as primary. Second, that are act on the non-additive secondary processes that are on the primary processes. So as I say, most of the methods are additive. They do not try to deconvolute the secondary processes from primary processes, and we can observe an effect of this. For example, in COSMIC, we have at least 7 signatures that are associated with DNA mismatch repair. And at the same time, this is a paper that's been mentioned already today. We, for example, know that there are 2 signatures, 14 and 20, that are both mismatch repair deficiency related, but the first of those signatures has Pol E deficiency in addition to mismatch repair, and the second has Pol D deficiency in addition to mismatch repair. So additive processes will multiply the signature, because for any combination of the signatures, primary and secondary, we're going to have and need to produce a new signatures. And also there's also additional inconvenience is that if we in those signature 14 and 20, even if we know that that there are a combination of polymerase deficiency, one of those polymerase deficiency and mismatch repair, we really don't know whether there is just one mismatch repair deficiency or there are various variants of mismatch repair deficiencies that will produce that will act differently. So this information cannot be obtained from this type of analysis. So in order to deal with this issue, we developed a method that allows for simultaneous modeling of primary and secondary mutational process. So in a way, our approach models this interaction and this dependency between those processes. So I'm going to describe our method, how it was originally designed. We obtained very interesting suggestions from the reviewers who actually challenged us to extend this method, and we did, but let me start with the simple design. So in our simple design, first, we assume that we have given the mutation matrix as always. In addition, I was avoiding this, but I have to tell it now. In addition to have a better model, we also assume that we can subdivide the genomic region into some local, some subregions, and those subregions might be related to dividing the genome regions into transcribed, non-transcribed, on dividing them into, depending on replication time, on defining them in another user-defined way. So what that cost us is that if we add this additional partition, we will have to infer the weight or the probabilities with each a given mutation hits in each of those regions, which of course assumes that those probabilities are not the same. So that's one input. The other thing we assume here is that we are getting all the primary signature as part of input, that we are not really trying to define them. We assume that they are given. We can drop this assumption, but I'm not going into this. What is our output? We computed the strength of those primary signature. We are compute those weight as green weight as needed since we introduced this partition. And then in addition, we have to infer everything about secondary signatures because we know nothing about them. So we have to infer the signature. We have to infer, of course, strength of the signature. And just as in this case of primary signature, we assume that those signature may have different strength in different regions. So we additionally infer those strength. At the end of the day, we end up to optimize this tensor equation. This first part corresponds to the generally modified non-negative matrix factorization with additional matrix W for the regions, and this is additional component from secondary signatures. Without going into more details, what did we learn? So when we apply this methods to breast cancer data, we're pleased to note that that previously proposed 3 different mismatch repair signatures disappear, and we just have one mismatch repair signature, and that signature is the same independently whether we chop the DNA into regions related to transcriptional processes or whether we chop it into regions that define different replication timing. And this signature is really very closely resemble the experimental signature obtained in Serena's lab. Okay, I'm running out of time. To summarize, I discussed 2 types of interactions between mutational signatures. One is interaction between mutational signature and molecular pathways. I show some of the application of this type of analysis. So we also use this approach to when we use mutational signature and expression data both from the same cancer, and then we try to understand the interaction between mutational signatures and pathway in molecular pathway in cancer. We are also working on inferring a different model. This clustering has always this disadvantage that we learn about a very broad generic pathway. Gone enrichment analysis would like to find some more mechanistic explanation. With respect of interaction between mutational processes, we now have a model that allows to model this composition between damage and repair. What we would like to do is to extend this model, because if we have those two proteins, this sometimes like in case of polymerase and mismatch repair, we know that they are together, but we really would like to know which came first. So we would like to be able, at least in some cases, to find the time dependencies, and we believe we should be able to make this type of extension. So I would like to acknowledge all the members of my group, and the signature decomposition was led by Damian Wójtowicz. The interaction between molecular pathway by Yuah Kim, and the analysis of cellular decomposition was done by Ermi Hodges. I would like to thank NIH Intramural Program for support. I would like to mention this one. So a couple of years ago, Ludmil and others organized this conference about AE and tensor factorization that I attended, and I think that was very helpful because it allowed us to have a very concise formulation of the molecular problem that we have, and through it to the tensor flow to solve it efficiently. So that was very helpful. And also our collaborators on our area study related to mutation signature, Mike Leifson, Roder Sharan, and Fabio Vandin. Thank you.
Thanks so much, Teresa. And I agree, I'm always in favor of another math workshop or conference. Happy to attend those. So we do have a couple of questions and a few minutes to answer them. So the first one is from Ludmila. And she asks, how do you evaluate statistical significance of those correlation coefficients? So it's about halfway through your talk, and apparently they were all under 0.25, given that just about 40 samples and multiple cell populations were tested.
So in the case when we are looking, so in the application, this application that I presented, because I thought, you might be at this COVID time, so it's an interesting application. We have small number of samples. So we keep every gene that has correlation as p-value of 0.05 or less. But because this is Gaussian enrichment analysis, even if we, it's okay that some of those are not so correct. For when we perform similar analysis in many cases for cancer, and then we have enough samples and we can be very strict about the p-values that we are applying.
Excellent. We have another question from Howard, and I hope I asked this correctly because I'm not sure I quite follow it. So he mentions AKT stimulation blocks FOXO1 phosphorylation and retains it in the nucleus. Mentions the previous speaker, I believe he's referring to GAD. Smoking nicotine stimulates AKT. Is this associated with FOXO1 retention in the nucleus?
That I wouldn't know. And I think it would be nice if we can discuss offline this question, how we can answer it. In general, we see perhaps that also I've seen some of discussion coming that how signature 5 and 40 differ from each other. I cannot tell much about signature 40, but signature 5 appears to be related to oxidation. So, and they co-occur with signature 4. But I would be very much interested in the specific questions that are just posted, although, not having sufficient biological background to answer by ourselves. So it would be nice if we can understand this question better.
I don't see any additional questions here. I do have one of my own though. Are you able to comment any on synergistic effects of these? You mentioned several times, and some of the others have as well, that these, we assume sort of additive mutations. Is that always a safe assumption? I'm sort of in the back of my mind thinking of, asbestos and smoking have sort of are believed to have a multiplicative effect, at least on risk. Does that extend down to the signatures?
So this is so far we looked most carefully at this composition of this mismatch repair processes and the damage. But of course, when we run our repair seq, we are getting also sometimes some other signatures, secondary signatures that are not being picked up by additive processes. Now, those we have to interpret. And one of the things that we seem to be getting is, for example, ApoBec. And in the context that, of course, for ApoBec, you have to have single-stranded DNA, and this may occur as the result of other processes, in particular homologous recombination deficiency, some topoisomerase deficiency. So then this signature might be picked up also as secondary signature. So this type of synergistic processes are being picked up, although mismatch repair is the one that is most easily validated and captured and interpreted. Mm-hmm.
Excellent. Thank you, Teresa. So we're going to transition over here next to David Wedge from the University of Manchester, and he'll be giving a talk on mutational processes and cancer evolution. And just give us a second to...
I'm still sharing. Okay. Thank you.
And I think David will take over, or it'll be handed to him by our facilitators. And again, I'll remind all attendees who are not hosts or panelists to please continue submitting your questions into the Q&A. It should appear at the bottom of your screen, a button that'll open up the Q&A and allow you to submit questions. All right, so take it away, David.
Thank you. I'd like to start by thanking Terry and Ludmil for giving me the opportunity to talk about two of my favorite topics, mutational processes and cancer evolution, and the interaction between the two. So the data that I'll be talking about today comes from the ICGC, the International Cancer Genome Consortium, which collected about 25,000 cancers from multiple different projects in different countries. 2,700 of these cancers underwent whole genome sequencing and were analysed by the Pan-Cancer Analysis of Whole Genomes Consortium, the PCOG consortium, which has been mentioned in some of the earlier talks. There was a number of initial analyses carried out on the 2,700 tumours. They were run through 3 analytical pipelines to form a consensus set, including SNVs, indels, copy number, and structural variants. Underwent QC, and then were then subject to a number of downstream analysis by several different working groups. So there were 16 different scientific working groups in total. I'm going to be talking about, as I said, the outputs from 2 of these working groups, the mutational signatures and processes working group and the evolution and heterogeneity working group. And I think some of the most interesting things to come out of PCOG were actually at the interface between different working groups. So, So cancer it's often said that cancer is an evolutionary process. This figure, taken from a paper from Lucy Yates and Peter Campbell from 2012, shows in a bit more detail what we mean by that. So the process of tumorigenesis, takes place in which the normal healthy cells become cancerous. That can be because of normal endogenous processes. It can be because of exogenous processes such as tobacco smoke. Then as tumours grow, they are subject to microenvironmental factors such as hypoxia and acidity. Tumours that may then undergo treatments, which can also cause signatures with mutations with particular signatures arising from chemotherapy or radiotherapy. Later maybe targeted therapy, and then when the tumours metastasize, they're subject to a very different microenvironment, and when they lodge in a distant organ, have to adjust again to establish themselves in that microenvironment. So what we wanted to do within the evolution working group, combined with the signature, the outputs from the signature working group in PCOG, was to really study this evolution from the point of view of signatures, and so I try and identify which signatures tended to occur maybe before tumours actually had become visible as tumours, versus those mutational signatures which were operative at later stages of tumour growth. So the first thing that we did was to separate the cancer evolution into 3 phases. So if we look at cancer genomes, then we see that that about almost 50% of cancer genomes have actually undergone whole genome duplication, which you can see on the bottom of this figure. So starting with just 2 chromosome copies, all the chromosomes are doubled, resulting in a tetraploid genome. Slightly over half of the genomes will remain diploid and won't undergo whole genome duplication, but those genomes that don't undergo whole genome duplication are likely to have gains of some chromosomes, so they will be likely triploid on some chromosomes. This gives us an opportunity to time mutations that have occurred within tumours, because if mutations have occurred before a chromosomal gain or a whole genome duplication, they will then appear on 2 chromosome copies, whereas if they occur after the whole genome duplication or the chromosome gain, they will only appear on 1 copy. And so by looking at the allele frequency of mutations, we can estimate whether they have occurred either before or after whole genome duplication. And we can also turn it the other way around, and we can actually time the copy number event itself by calculating the ratio of mutations that are on 1 copy versus the number that are on 2 copies. We can actually time in molecular time the occurrence of the whole genome duplication. Also, by looking at the allele frequencies, we can distinguish clonal mutations, which are those which are present in all of the cells in a tumour, and subclonal mutations, which will be only in a fraction of the cells in a tumour. And again, this feeds into our evolutionary analysis, because we know that the clonal mutations must have occurred before the subclonal mutations. So dividing our cancer evolution into 3 epochs, which we define as early clonal, occurring before chromosomal gains, late clonal, which means occurring after chromosomal gains, and subclonal, meaning that the mutations which are not present in all cells in the tumour. We were able to, looking across all the different tumour types, identify whether signatures had a tendency to occur early rather than late, so before or after whole genome duplication, or to occur either clonally or subclonally. I'm going to particularly focus on the early events to start with. So the red box shows the signatures which were significantly more likely to occur before whole genome duplication, so they occurred early. And these are, you can see the signatures. So we have SPS7, which is associated with UV light exposure, and nearly all of the tumours in this category you can see are black dots, which is melanoma. SPS12, which is found in almost exclusively in liver cancer, but has unknown etiology. SPS4, which has been mentioned earlier, is associated with smoking. SPS1, which is the clock-like process which occurs in normal tissue, and so we would expect that to be an early signature, because many of those mutations will have occurred in normal tissue rather than cancer tissue. Similarly, signature 5, and SPS17, which is seen in mainly in esophageal cancer, and all of these signatures are seen to occur generally early and before most copy number changes. Moving to the other end of the spectrum, there are a number of signatures which were significantly likely to occur after chromosomal gains. These are, you can see, SPS3, which is associated with double-strand break repair deficiency, and this we think the reason this appears late is because in most cancers the the homologous repair defects will have occurred, appeared somatically from somatic mutations in BRCA1 or BRCA2 or other HRD genes, and so that signature 3 will appear after acquisition of HRD. Other signatures that are seen late are the APOBEC signatures 2 and 13, which has been previously reported, and double base signature 5, which is associated with platinum chemotherapy. Moving to the bottom panel, where we distinguish mutational signatures which occurred in clonal mutations versus those that were subclonal. We identified SBS9, which is quite interesting that it tended to occur early and clonally. I'll say a bit more about that later. And also indel signatures 1 and 2, which are associated with slip strand mispairing and leading to mismatch repair defects. These again tend to occur early and clonally, and in tumours that have mismatch repair defects that are hypermutated, and we tend to find that in those hypermutated tumours, the great majority of mutations are clonal rather than subclonal. So that was a kind of a high-level view, looking at the kind of aggregate of all of the samples within each tumour type. But we can zoom into individual tumours as well, and trace the trajectory of mutations within individual tumours. So we first ordered all of the mutations within each tumour from early to late, and then we searched for breakpoints in the signatures which indicated there'd been a substantial change in the mutational processes. These are shown as the red vertical bars on the 4 samples on the left, represent these breakpoints between different mutational signatures. We can then combine these signatures across the different samples from different tumor types, and what we observed was that there was significant change in these signatures that I've marked up on this slide. So SBS9, which I mentioned earlier, is seen to occur commonly in CLL, chronic lymphocytic leukemia, and also in B-cell non-Hodgkin lymphoma. Interestingly, these tumours originate in post-germinal centres in B cells, which then migrate from the germinal centres elsewhere. And it's thought that this signature occurs in the B cells, and then but then not later on. So we can, I think, explain why this signature tends to be early. And you can see SBS9 is very high early on in those tumour types, and then drops off at later stages. The other signatures the next 2 I think I mentioned before, SBS4 in lung cancer tends to occur early, SBS17 in esophageal adenocarcinoma, and also in thyroid cancer, we tend to see APOBEC signatures 2 and 13 actually tend to occur at a later stage. So they have low prevalence at the early stages and appear much stronger at the later stages. So we can, this is a kind of a summary of what I've shown you in some individual cancers, showing you across all of the different cancer types. We can identify a number of the features that I've just mentioned. So we can see again increased signature 4 tends to be clonal and early and on the left, whereas and also in melanoma, signature 7 tends to be early, and in esophageal adenocarcinoma, signature 17 tends to be early. I'm just going to go back because I actually forgot to mention something on the last slide. So we compared the trajectories in terms of signatures with our identification of subclones. So we clustered all of our mutations into clonal and subclonal clusters. In some of our tumours, we were actually able to identify multiple subclones within our tumours. And then we compared our switch points in the signature analysis with the switches between subclones that we observed from our analysis of the allele frequencies. And what we observed was that the breakpoints that were close together between the signatures and the subclonal analysis were enriched. So we saw the breakpoints that we observed in the signature analysis tended to be close to breakpoints that we identified from the allele frequency analysis. So we saw in the real data, we saw more breakpoints that were close together than from random simulations. We also observed that in samples where we identified more signature change, a greater percentage of signature change, they tended to have also more subclones. So you can see in this figure E that the samples that are shown in kind of orange were those that had no subclones, and they tended to have fewer changes in signature. So again, the two analyses support each other. The samples where we saw most signature changes also were the ones where we observed more subclones from the subclonal analysis. And the final two figures on the bottom right show you first the overlap in the breakpoints that we identified from the two analyses, which was quite substantial, but we also identified some additional breakpoints from the signature analysis. So I think this tells us that actually we are, from our subclonal analysis, we're not actually identifying all of the subclones, and actually by adding in the signature analysis, that enables us to identify further subclones that were undetected in the standard subclonal analysis. So I think signature 1 has been mentioned before, and it's been previously reported, but we kind of replicated the analysis that was done previously by Ludmil Alexandrov on this larger sample set, and we observed that signature 1 correlated with age at diagnosis in just about all of our tumor types. This means that we can use signature 1 to actually time events in real time. So we looked at two different events. The one on the top is whole genome duplication, which I mentioned earlier. So we can count the number of mutations on one copy and on two copies, and from that we can time the occurrence of whole genome duplication. You can see what we found. So across different tumor types, we observed that the whole genome duplication had occurred some years prior to diagnosis. And if we look on the right-hand side, in ovarian cancer, the whole genome duplication often had occurred up to 20 years or even more prior to diagnosis. The second event that we timed was the appearance of the most recent common ancestor, after which we see heterogeneity start to appear within the cancer genomes. The appearance of heterogeneity we timed to be up to about 5 years prior to diagnosis. So we don't see as much latency in terms of the appearance of the most recent common ancestor, but we do see that there have been several years in which the tumours have continued to evolve and develop heterogeneity, subclonality. So far, I've mostly been talking about SNV and indel signatures, and we were also interested in copy number signatures and structural variant signatures in kind of wider sense. So this figure shows you the timing of chromosomal gains in different tumor types, and we've separated it by different chromosomes as well as by different tumor types. The purple colouring indicates late events, and the green indicates early events. So these are ordered by tumor types which tend to have copy number gains early at the top, to those that tend to have copy number gains late at the bottom. I'll just draw your attention to GBM at the top. So these tend to have very early copy number events, but also they tend to occur on particular chromosomes. So you can see that we very commonly get chromosomal gains on 7, 19, and 20, and they are very early events, suggesting that they may be drivers of tumorigenesis. I'll just mention one other or two other tumor types, which are different types of kidney cancer, the papillary and clear cell renal cancers. And what we observe here is there's a really enormous difference in the copy number profiles of these two different subtypes. So in the papillary cancers, they tend to have more copy number gains, shown by the larger circles, but those copy number gains tend to occur late and so they appear in with the purple shading. So we can look at the different chromosomal gains that have occurred within the genomes, and we can see whether they tend to occur at the same time or at different times. We observed in our samples shown on the left-hand side of the pie chart are the tumours that had undergone whole genome duplication. And we can see that a large number of them had occurred synchronously, which is what we would expect. If it's a whole genome duplication, we expect that the different chromosomes have all been duplicated at one time point. But very interestingly, when we looked at the samples which hadn't undergone whole genome duplication, we observed that actually a large proportion of those had also occurred synchronously. So it appears that we have mutational processes which are occurring, which are leading to multiple chromosomal gains on different chromosomes. I'm going to speed up because I think I'm kind of running out of time. But I'm just want to kind of briefly show you this landscape figure. So this shows you all the different mutation types, the SNVs, indels, copy number, and SVs. The point that I want to make here really is that these samples are tumor types are ordered by the level of heterogeneity in the SNVs. So you're getting increasing heterogeneity as you go from the left to the right. But when we look at the other types of aberration, you can see that they don't go in the same order. So we have some tumor types which tend to acquire SNVs early and clonally, but then acquire copy number late. We have other tumor types where actually they occur in a different order, suggesting that that some tumor types may be more driven by copy number changes at the early stage, and then they acquire SNVs at a later stage. I'm going to skip through those. I'll just finally say we're also over to time complex punctuated events, including chromothripsis, chromoplexy, and catesis. We observed that catesis tended to occur late across multiple different tumor types. This is shown in red. It tends to be below the line, indicating that they tended to occur late. Chromothripsis was actually more likely to be early, shown in blue and above the line. And we think this is probably because chromothripsis is more likely to act as a driver, because it really has a massive impact on the fitness of the genomes, whereas the majority of catesis actually occurs in non-coding regions and is likely to be passenger events. I'm going to skip over the rest of the slides and just thank everybody who contributed to this study, the patients and their families, and our funders. And thank you, and I'd be happy to take questions.
This is fantastic, David. Thank you. So I see we have two questions, one from a colleague at NCI, Mike Espy. He asked, were you able to show unique signature change trends tied to specific therapeutic regimes, or also pre-therapy signatures that could be tied to those that appear with therapy resistance? And he mentions intrinsic versus acquired.
Right. Yeah. So we did observe in patients that had received platinum therapy. Yeah, we observed the onset of the platinum signature, and that tended to occur late. And we could see that in individual patients. We could see the increase in that. I think that the number of samples within our cohort, I'm not sure that was enough to look at the if there were differences in the tumours that acquired treatment resistance. I'm not sure that we had the numbers to look at that within this study.
Dimitri has a question similar to mine, actually. He asked if you confirmed clock-like signature for SBS5 or just for SBS1. And then I had a question about what's the time resolution of these clock-like signatures in general, or is it sort of an aggregate effect of them?
Yeah, so the signature one, we had a clearer signal and a stronger correlation with age, and we also looked in some tumours where we had multiple samples over time, so we were actually able to test that hypothesis. So we saw that signature one it correlated very well with time. Signature 5 was variable across different tumor types. So we found that it correlated better with time in some tumor types than others. I think there are a couple of problems with signature 5. I think it may not be as clock-like as signature 1, but I think there's also some difficulties in deconvolving signature 5 because it's a much flatter signature than signature 1, much less distinctive, and can get confused with signature 40 and signature 3 and other signatures.
You can push us another 60 seconds, Dimitri, if you want to follow up there.
No, I just wanted to show my face while this question is answered. Thank you.
Yeah, you asked about the resolution. I mean, I think we've seen that the clock-like signature 1, although it does occur at roughly clock-like rates, it occurs at a different rate in different tissue types. So I guess that is the limiting factor in terms of the resolution.
Okay, thank you. All right, so next up we have Peter Park from Harvard University, and he's going to speak on algorithms in mutational signature analysis, SIG-MA and MUSICAL. So we'll give Peter a chance to start up his presentation, and as soon as you make it full screen, I think we'll be all set. Excellent.
Excellent. All right, well, I'm delighted to be able to tell you about two algorithms that we've been developing in the lab. Well, given what you've heard already today, I don't need to tell you the basics, except to remind you that there are actually two main components to the signature analysis. The first is the de novo discovery part, where you have the mutation count matrix that's decomposed into a signature matrix and exposure matrix. We'll just use the notation W and H for the signature and exposure matrices. And then there's a refitting step where once you have a catalog of signatures in an individual genome, you like to calculate the exposure of different signatures. The two questions that we've been asking in the lab are the following. Can we detect mutational signatures from a small number of mutations, for example, from panel sequencing data? And I'll tell you why this is such an important question in terms of its application to the clinic. The algorithm itself was published 2 years ago, but I will describe the main idea and then tell you about the validation data that we've been getting from our collaborators. The second part will be just more generally, how can we improve the accuracy of the analytical pipeline overall? And there are three ideas that I will outline when we get there. And this is wrapped up in a package called MUSICAL. The work that I'll describe have been done by Doa and Hu. Doa developed Sigma, and then for MUSICAL, Hu and Doa have been working together. Our motivation for identifying mutational signatures from panel data is signature 3. Of course, it's not confined to signature 3, but let me use that as an example here. The link between signature 3 and homologous recombination deficiency has been made by many people for many years, so I don't need to go over that. There have been experimental validation as well. So for instance, if you knock out genes in HRD pathway, you get profiles that look like signature 3. There are other signatures of HRD as listed there, but for the purpose of identifying signatures from panel data, we're going to talk just on point mutations because the other features are a little bit harder to understand. You already heard from, even from David Witch just now, as well as from the earlier sessions, that these flat signatures are relatively hard to work with. And signature 3 is one such signature. Why do we want to detect HRD from mutational signatures rather than, say, BRCA1-2 mutations? Because there are many ways in which HRD can arise. In addition to point mutations for BRCA genes, you can have DNA methylation of BRCA genes and many other pathways listed here. And so by identifying based on mutational signatures, we think we can define a larger set. And as you know, PARP inhibitors would be a potential treatment for that. If you have whole genome or exome data, HRDetect and other algorithms are available. HRDetect is from Serena's group in 2017, where both signature 3 and other rearrangement and other indel signatures were used. But here, the question is, could we detect signature 3 from panel sequencing data? Now, why do we want to do this? Well, as much as we like to work with whole genome data, there are a lot more panel data. And so I listed 2 examples here. The Project Genie is a collaboration between multiple hospitals, including MSK in New York City and Dana-Farber and many others. Then, of course, there are commercial platforms. As much as we like whole genome data, panel sequencing will not go away. And that's because for many clinically relevant mutations, you do need such high coverage. So 1000x plus. We've done some careful analysis of exactly what the relationship between specificity and sensitivity is with sequencing coverage. It's described in this paper from 2017. But basically, we'd love to be able to detect signature 3 from panel, also because it obviates the need for additional testing. So from a clinician's perspective, if you suspect HRD, you could order this $4,000 assay. But if you're doing panel sequencing anyway, you can get this information for free. So the mathematical question then is, well, is this doable? Here I show what signature 3 looks like from whole genome data. Below, I show what it would look like if we were to sample from the targeted region in a panel. And when I first looked at this problem, I thought this was not possible. You just don't have enough mutations. There are some tricks that we do. Obviously, we want to use both non-synonymous and synonymous mutations, but then we actually, ideally, would get the BAM files and realign and get those reads that map to off-target regions. We can be a little bit lenient on the calling criteria because mutational signature analysis is somewhat robust to these additional false positive calls. But basically, we have at least 1,000-fold reduction in data. And so how do we do this? Well, let me just describe the 3 ideas. The first idea is that we make use of previously collected data. If we didn't have PCOF data, we would not be able to do this analysis. And what these data, whole genome data, allow us to do is we know the co-occurrence patterns of different signatures. So we have much better sense of what we expect. The second idea is to use a likelihood-based matching of spectra. Most of you are familiar with cosine similarity, and cosine similarity works fine if you have a lot of mutations. But if you think of them as vectors in space, once you have much fewer point mutations, these become unstable, and your assessment is not as good with cosine similarity. And we think that a likelihood-based comparison of spectra is a much more natural and accurate approach for this case. And then we have a more robust prediction framework. We use something called gradient boosting to combine data from different mutations to come up with a prediction scheme. Indeed, when we call signature 3 positive cases, only just over a quarter of our samples have BRCA germline or somatic mutations. The rest we would not have detected if we are looking just for BRCA mutations. Now, on the right, these are estimates from the paper on the prevalence of signature 3 cases. We have we have a collaboration with at least a dozen clinicians. Some of them, especially in cases where the frequency is not very high, are very excited about the possibility of detecting HRD from panels because, say, in pancreatic cancers, if you know that there are only 5% of the cases that have HRD, you're not going to order a separate test. And so I think this is a very important clinical problem. Let me share now some data from our clinical trial collaborators. On the left, we show that if we were to sample from TCGA data, BRCA mutant cases here in blue are actually quite similar to the signature 3 positive but BRCA wild type cases in terms of survival. And then the signature 3 positive and the negative cases are quite separate. If we look at OncoPanel data, this is the platform used at our local collaborators, Dana-Farber Cancer Institute. Again, we see that the signature 3 positive and negative cases are separated in terms of progression-free survival. We also looked at a trial called TOPACIO. This is a PARP plus PD-1 inhibitor trial for ovarian cancers. And here we see that there's a nice separation in terms of survival. In this particular trial, they actually had multiple assays to assess HRD. It turns out that Sigma outperforms all of them in terms of showing significant difference. The expensive assay that I mentioned, near my choice, does not show significant separation. Just two more data sets. So one is a PARP inhibitor olaparib trial in combination with PI3K for breast and ovarian cancers. Again, signature 3 status appears to be a great separator, whereas other covariates, including BRCA, germline, or other genome instability score, which mimics this myriad platform, does not do that. And finally, for the Peter Mac trial for PARP inhibitors in breast cancers, we see that signature 3 captures not just the ones that are detected by gene level analysis. So these are BRCA mutations in purple, germline, somatic, or amplification or methylation, which would be captured by a good panel. But it captures these cases that have DNA methylation of BRCA1. And if you look at objective response rate, there is a pretty high response rate for the signature 3 positive cases, not dissimilar from what you get from BRCA1 germline cases. So that was just a quick overview of the data sets that we've been working with. I hope this is good evidence that our methodology is working. Now I'd like to switch gears and talk about this other effort that we have. The question here is, could we improve the accuracy of signature analysis overall? This morning's talk from Dr. Landy provided a wonderful introduction to this, because she pointed out discrepancies between the results from different tools and many other issues. This package has multiple interesting features. Let me start with this minimum volume NMF idea. So some of you who remember linear algebra know that we can think about a geometric interpretation of these vectors. So here we have 3 signatures, signature A, B, and C, that span the data point. So we can actually simulate these points using the 3 signatures. When we use NMF to solve for the solutions, it turns out that the solution is not unique. It's because there are multiple these dots that can span your data points. And so you know that when you run NMF, you'd have to do it multiple times, because every time you get slightly different answers. What minimum volume NMF does is to penalize this value, right? So this is an additional constraint so that the dots, the 3 circles that span with a minimum volume is the solution that you get. This idea is not ours. This has been around in different communities for a while. The earliest references that we can find are listed there. Let me illustrate the advantage of using minimum volume NMF with this simple example. So suppose you have these 4 signatures. We use random exposures of random weights of these to simulate the data. And so in this PCA space, you have the true signatures and then the samples generated from these signatures. If you use NMF, every time you run NMF, you're going to get a slightly different result. You're going to capture the 3 signatures down here pretty well, right? But for the flat signature, you get slightly different answer every time. And so it's shown on the right. The first one kind of looks like SPS3, but not quite. When you do the standard refitting step, what happens here? It looks for the combination of signatures that matches this. And what happens here when you don't have the right signature here is that it will introduce artifactual signatures. So in this case, SPS 39 gets brought in this NLS step. And that's what we like to avoid, because you're going to get lots of false assignments. And so if you use minimum volume NMF, in fact, you recover SPS3 exactly. as illustrated by the red dot here, a red triangle set there. So that's the first step. The second part is for improving non-negative least squares refitting step. So let me illustrate this with a simulation. Suppose you have these 7 signatures. With these exposures, we simulate a sample. A standard NLLS algorithm will generate a bunch of signatures with these weights. You can see that it has both true signatures and some false signatures, because what this step does is to find a linear combination that best explains that with some minimum error, this observed sample. And you can see here that obviously you're over-assigning these signatures to the sample, and so you like to avoid that. And so most algorithms will try to make it sparse, that is to bring the coefficient to 0. And one simple way to do that, of course, is to just have a fixed threshold, 5% threshold for this illustration. But you'll see that it will still capture these false signatures. Okay? And so what we propose here is use likelihood-based non-negative least squares. And let me illustrate on the next slide how well this works. But we think this is a much better approach. So here, it's the same simulated case. The fact that we use likelihood-based approach does not mean that we don't have a threshold. We still have a threshold that we need to tune. I'll get to that a little bit later. But if the threshold is high, we capture the most significant one here, so SPS 5. As you turn down the threshold, you're going to capture more of the true signatures. If the likelihood threshold is too low, then you're going to get the false signatures as well. So it's important to be able to find the right threshold. Now, if you were to do cosine similarity for this example, there's no way that you can recover the true set of signatures. There's no continuity here, as you see in the top plot. So we think that with a properly chosen threshold, this likelihood-based sparse non-negative least squares is a good approach. Now, for the final bit that I like to describe, let me illustrate by this example. Suppose we have these exposures from 720 whole genome data. Each column is a sample, and each sample consists of multiple signatures shown here. And then we know what the signatures are. And so suppose we simulate the data using some multinomial sampling of these cases. Okay. If the process of obtaining signatures, discovering signatures works right, when we perform NMF, we should get the cosmic signatures back. But in fact, that is not what happens under standard NMF. So of course, we have this for all signatures, but I'm just showing you 2 examples here. For signature 1, in fact, when you reverse engineer this, then you get a signature that sort of looks like signature 1. You can see that the errors are pretty small. But for a flat signature like signature 3, you get errors that are on the same order as the signature itself. So this is not reproducing the signatures correctly. And intuitively, if you just do this process of simulating data from your decomposition and then redoing the decomposition, you should get something similar each time. That is the idea underlying our approach here. And so you, given your data set, do the decomposition, matched catalog, refit, and signature assignment. And then we're going to generate sample from this distribution and generate a new count data, and then decompose that matrix. And how this original W and this W is similar is our measure of consistency. And we can do that for the exposure matrix as well. And so we can, in fact, use that idea to tune parameters. And so we can optimize doing a grid search of the parameters until we have a pretty good consistency between these two. Well, it turns out that this approach works really well. here's an example of glioblastoma data. So 39 samples, we do de novo discovery, and we get these 4 signatures. And then we use this likelihood-based matching for matching to catalog, and then refitting, and then we get this set of exposures. And so that we have 6 signatures and their exposures. Now, this is quite different from PCOG results. So when we look up PCOG data, this is what we get. We get the infamous signature 40 here. And I'll show in the next slide that there are the differences in the W and the H matrices. But we think that signature 40 is real, especially for kidney, uterus, and prostate, but we think that it's an overassignment in many other tumors. Well, how does it work if we actually look at the W matrices? This is what we get from the data. We get from Musical, we get from PCOG, and we see that for some signatures, it's not so bad, but for signature C, for instance, you're not capturing the right signal here. And so the error is higher for the PCOG set. If you look at exposures, same problem. We think that the exposure is much, the exposure matrices are much more consistent with the Musical derived H. Now we've done this for PCOG data set as well. I don't have time to tell you the details of this, except to say that in a similar way, the errors are smaller for Musical across these different tumor types. The red bar you see is signature 40 that we think is overassigned. Indels, we identify some novel signatures. And then we have some examples of what the reasons to think that this is a little bit better. Age and clock signatures, for instance, are more consistent across data sets. So hopefully we'll be able to publish this paper soon, and you'll get to read the details. And just to summarize, with Sigma, we think that it's possible to identify signatures from panel data. It's done in a tissue-specific manner. The more data sets we have from public repositories, the better it's going to be. We are happy to assist people in implementing the method for hospitals. We don't have enough hands at the moment to help with all the inquiries that we've been getting, but hopefully we'll get some resources to that soon. With Musical, we think that some of the innovations you can take and implement within your favorite framework. We think that this approach does not require manual curation, as it sometimes does, in picking the right signatures for different tumor types, and allows for full reproducibility. Again, Doa and who did all the work, all the clever ideas are due to them. This work was funded by Ludwig Center at Harvard, as well as Cancer Research UK, and we're looking for other people to join us in our effort to push the boundary on the signature analysis, and especially as we try to apply this to the clinic. Thank you very much.
Thanks, Peter. We'll do a couple quick questions. Dimitri has a question that then leads into a question that I have actually for you. So he mentions that a striking feature of the SBS3 signature is a clear depletion of G following mutated C. Otherwise, the signature is rather flat. So he asked if you could use this G depletion as a simple statistic instead of the 96 channel SBS3 signature.
Well, I think there are many signatures. I mean, you can look at signature 1, and I think there are some signatures where you could reduce it down to a smaller set, but I don't think there's any problem in this framework of just expanding it to the whole set, because all the other signatures are using the full set. So I guess, Dimitri, the answer is maybe, but I'm not sure whether that's particularly helpful in the whole framework.
So the follow-up to that is, Peter, my first grant at the NCI was a data visualization grant that you held with Niels Gellenberg. So I'm going to ask a data viz question. So as we point out here, these trinucleotide context signatures, have 96, essentially a 96 bar chart. What are we going to do if we go to pentanucleotide context? How are we going to visualize? And especially when you have these sort of, combination signatures with that's including indels and chromatosis and other things like that. What and who's working on that, and what do you think there's a solution in sight?
I don't have a good answer to that. I do think that we need something clever, something interactive, so that the right part of the data are featured for the user. But I think a general problem to be solved.
Okay, I'll move over to the Q&A here. I want to make sure I understand this. So this is from an anonymous attendee. This was earlier on when you were speaking. From the Sigma tool, what parameter would be more appropriate to study gene association with SIG3? And they mentioned exposition to SIG3, SIG3 ratio, or SIG3 MVA.
I'm not sure how to answer that. I think it would depend on the exact analysis. I'm sure Dora would have some ideas too. I think we'd have to really just check to see what kind of association you're trying to make with using what data. I think any of those could work, but we'll have to test them out.
Okay, great. Well, we'll switch over to the next speaker here. And as Josh comes along, actually, you know what? While Josh comes up, maybe we do have one more time for one question. So in comparing musical with PCOG-W, you could exclude SBS 40 using musical, but SBS 5 was still very frequent in the musical assessment. So Terry asks, doesn't SBS 5 have similar issues of SBS 40? We are quite in the weeds of these signatures already.
Yeah. So, I mean, SBS 40 is not an artifact, we don't think, because it is quite strong in kidney, for instance. And so, SBS 5 is aging associated, and we do see it. And SBS 4, we see it across different tumor types, and 40 is specific. And so I don't think that is a problem. They're relatively flat, but they're not totally flat. And so they do have some information. Okay.
Oh, yeah, it looks like you actually have to stop sharing so the next person can. So I didn't realize that actually, sorry.
Thank you for the questions.
Sure thing. So I'm excited to bring up another one of NCI grantees within my portfolio, Joshua Campbell at Boston University, and he's going to give a talk, Opportunities and Challenges with Bayesian Modeling for Mutational Signature Inference.
Great, awesome. Thanks, Dave, so much for the introduction, and I'm excited to follow Peter and actually start addressing one of the questions that you had. So I'm an assistant professor at Boston University, computational biologist. I'm working with several colleagues in the mathematics and statistics department. We're all interested in applying Bayesian methods to help improve the process of mutational signature inference. Let's see, start up my laser pointer. Okay, there we go. All right, so since the very beginning, the first paper by Alexandrov and colleagues, they used the non-negative matrix factorization, a method which we've named over and over again today and are all very familiar with. And of course, since then, it's been used widely across many different publications and been one of the major workhorses. But I think as we continue to do this type of analyses, there are potential weaknesses that people are finding. Many of them have been discussed today, and people are trying to figure out ways around them. Some of the limitations of NMF are that it can be difficult to include additional information in the deconvolution process. There is no inherent method for predicting activities in new samples giving an existing set of signatures, and Peter covered that really well in the previous talk. But it can also be potentially sensitive to perturbations or noise in the data. So from my talk, I'll be describing a few different models that we're working on developing. The first one is a model that incorporates additional flanking sequence information into the deconvolution process, the signature generation process. Second one is a robust Bayesian model that simultaneously discovers signatures and predicts exposures of existing signatures from the database. And then in the very end, I'll review some novel software that our group has recently developed. So what other approaches are there? When NMF was being developed, or a little bit before then, There was another famous tool that was developed and widely used in the text mining field called latent Dirichlet allocation, or LDA. It's a similar idea, similar concept to NMF. In the same way, NMF identifies signatures by identifying co-occurring mutation counts across tumors. LDA was used to identify these topics or create these topics by identifying co-occurring word counts across documents. It was widely used in this field, but I think one of the most interesting things that came out of it was not the algorithm itself, but how people began to expand the algorithm and do various things with it. And this is one of the benefits of using a Bayesian framework, is that you can start to do additional things. So people started incorporating supervised labels, or incorporating sample level covariates, or user tags from text data, for example, to help in the deconvolution process. So what I'm going to show you in a little bit, we've been developing a model that helps capture additional flanking sequence information beyond the trinucleotide context. So what evidence is there that this information, that there is biases beyond the trinucleotide context? I mean, discussed it a little bit in some of the previous talks, but I actually really like this figure from this paper by Dateland et al. In Nature Genetics, from the Sunayev lab. And they looked at the extended context around particular mutations in particular tumor types, and just looked at the distribution of bases. And if you just, for example, look at all C nucleotides in the human exome, you see a pretty flat distributed, flatly distributed distributions for most of the bases. However, when you start looking at specific mutations and specific cancer types, you start seeing biases. So for example, if you look around C to T mutations in melanoma, you can look at the first upstream base to the left, and you see a strong prevalence of Ts at that position. But even if you go beyond that, you can still see biases in Ts 3 or 4 bases in. So why does NMF have trouble, or why is it difficult to capture this information with NMF? And it's because traditionally we have to predefine our motifs and count them ahead of time. So in the standard model, where we have, we look at the trinucleotide context, and we have 4 bases to the left and 4 bases to the right of the mutation, and 6 mutation types. That gives us our nice bar, 96 mutation contexts with our nice bars. And we mentioned earlier, maybe we can go out 1 base to the left and 1 base to the right and get 1,536 motifs. And Gadi talked about that a little bit earlier in his talk. And if we have whole genome sequencing data, enough counts, then maybe we can still estimate it there. But it does become a challenge to sort of visualize and understand the patterns that are going on. But if we even want to go out beyond that and extend that, it becomes even more difficult. So if we want to go 1 more base to the left and to the right, this would be over 24,000 motifs. And that would be just too computationally infeasible for NMF to be able to estimate it, and just the number of counts in each entry would get super low. So we can use LDA-like models to try to incorporate this information. I want to point out one previous piece of work by Qureshi et al., where they did a similar LDA-like model. They used the 6 mutation types as their central motif, and then went out 1 base to the left, or sorry, 2 bases to the left and 2 bases to the right, 1 base beyond the trinucleotide context. And they were able to do this deconvolution process. So they were able to identify different signatures and see biases in the bases beyond the trinucleotide context to some degree. However, the challenge here was using the 6 mutation type context. It can be a little bit difficult to translate it back to the trinucleotide context. And there's a lot of interactions that are happening, which are really important, and which we're all used to seeing to identify the signatures. So to help solve this problem, we developed a similar model, or expanded it, where we modeled the central mutation using the 96 motif schema. And then we went out 5 bases to the left and 5 bases to the right on either side, where we characterized the distribution of ACTs and Gs at each position in each signature. And I'm going to take you through a couple of examples showing you the patterns in the sequence context beyond the trinucleotide context. We actually used, just as proof of concept, we actually used whole exome sequencing data from the TCGA to do this. It's just to demonstrate that you can identify these patterns with lower numbers of counts. You don't need huge numbers of counts from whole genome. So the first signature I'll show you, we applied it separately to each tumor type. And the first signature I'll show you is the POLE one. So SBS10 from Cosmic V2. So you can see this similar signature was estimated across the 3 different tumor types. And if you look at the central part, that's showing you the distribution of the mutation contexts in the trinucleotide space. But then if you go now to the left or to the right, we can see the distribution of the different bases, the 5' or the 3' end. So for example, we can look up here and see that there's a very high prevalence of T's at the second position in the 5' context. And this bias actually can extend pretty far out. So even if you get up to position 4, for example, we can see a higher prevalence of A's. And even maybe a slightly higher prevalence of T's at position 6. And then we can also observe similar types of biases on the 3' context as well, where we see a higher prevalence of T's and maybe a depletion of G's. And what's interesting is that if you look across the signatures generated from different cancer types, we can see similar patterns suggesting that it's robust and can be found across multiple tumor types. So another signature that we looked at was ApoBec, and we looked at the 2 different ApoBec associated signatures in a few different cancer types, so SBS2 and SBS13. And again, we found signatures with high cosine similarities to it. We could see in SBS2 a slight bias for the pyrimidines, C's and T's at the position in the 5' end. And even in SBS13, we could see a slightly higher prevalence of C's compared to T's. And then if we look at the 3' end, we can actually see maybe some more differences between the 2 signatures. We see a slightly higher prevalence of G's and the 3' end for SBS2, but SBS13 actually has a little bit higher prevalence of A's. So just showing that even related processes can still have different patterns in there and the context beyond the trinucleotide context. If we move on a little bit more, and a similar idea we can find in with the UV signatures and melanomas, we could identify 2 distinct UV-related signatures. This time we're correlating with Cosmic V3, and so we could see SBS7A and SBS7B. I think what's really interesting here is that 7A, we can see a very high prevalence of T's, like we observed earlier when looking at the whole cancer type, but we can observe them both in the 5' and the 3' end. However, in contrast, the SBS7B signature tends to be a lot more flat and even maybe has a slight depletion of T's at certain bases. Lastly, I'll just show that if we revisit the POLE signature in some of our cancer types, if we allow a larger number of signatures to be estimated, we can actually split up the POLE signature into the SBS 10A and 10B signature. Again, we can observe this across multiple tumor types. And we can see, if we look maybe on the 5' end, similar patterns, similar distributions for both those signatures on the 5' end. However, if we go to the 3' end, we can see that, for example, 10B has a higher prevalence of A and the 3' base, whereas the signature 10A has a higher prevalence of T's at several positions on the 3' end. So it's showing you that you can have these specific interactions between specific trinucleotide contexts and bases beyond that. So that was one example of how we can incorporate additional information using this Bayesian approach. I'm going to move on and talk about a second model that we're developing where we're trying to allow for joint estimation of known signatures and identification of novel signatures at the same time. So there's generally two different workflows that people try to follow, which have been pointed out a few different times across different talks earlier today. So you can have mutation counts, and you might want to perform discovery using something like NMF, where you identify both your signatures and your exposures or activities. But to then really identify what your signatures are, you would want to take those and correlate it to Cosmic or some other database. And maybe several of your signatures, you get a high cosine similarity, so you can confidently say, okay, this signature is that, and give it a name. But maybe for some of the other ones, especially the flat ones that we've been discussing a lot, signature 3 and signature 5, maybe you get a more moderate cosine similarity, and so you don't know what to call it. Maybe you just don't have enough samples or lower numbers of counts, and so this correlation becomes a bit weaker. Also sometimes this process can miss known signatures. It may have less sensitivity, especially if you have a smaller number of counts or lower number of samples. The prediction methods, and there are several out there, including Sigma, which Peter just told us about, takes into account pre-existing signatures from Cosmic and tries to estimate the activities for those signatures. Of course, there the question becomes, are we missing novel signatures? So then usually people, especially if they have a new cohort of tumors, they perform both analyses to try to get a sense of what's in their data. And so we're working on developing a model that can do both at the same time. And so we're using a Bayesian Poisson NMF model that's been modified in several ways. And I'm working with a talented grad student, Hao Yu Fang, and a collaborator, Jonathan Huggins, in the math stats department at BU. They actually had a poster on this earlier today, so you can reach out to us if you want more information on it or want to see the poster. But as a proof of concept, we generated a synthetic data set which had 12 signatures in it. And when we ran the model, we fed the model 8 of those signatures, and we left out 4 other signatures. And then we also gave the model all the rest of the Cosmic signatures that weren't a part of the data set. And so here is the part of the primary results, and each bar is showing you sort of the estimated mean signature activity for each signature. Generally, if the mean signature activity is estimated correctly, then the individual signature activities for individual samples are also estimated pretty well. And each asterisk is showing you what the true mean activity is for that signature. One nice thing that I'll point out about Bayesian modeling is the ability to quantify uncertainty. And so in this case, we're showing you the 95% credible intervals here, and you can see that a lot of these bars capture, the true mean activity of the signature. One of the things that Jonathan developed was a novel sparsity reducing prior to try to turn off signatures that aren't actually present in the data set. And you can see here working pretty well, turning off signatures 4, 6, 7, and 8. So this model also has the ability to estimate signatures that weren't fed to it. And so here you can see it estimated the 4 signatures that were left out. And over here you can see the high cosine similarities between the estimated signatures and the true signatures. I'll point out one interesting thing about the model is that signature 5, even though it was fed to the model actually preferred to turn it off and re-estimate it over here as a novel signature. So even though the model was correct and got all the signatures at the end of the day, it sort of switched it to a known one. So I think this is still an interesting problem and both a statistical challenge as well as a potentially biological one. How similar does something have to be to call it known or novel? And we're currently testing out different ways of introducing an error term that allows some variation in the given signatures. Along those lines, I just wanted to give a note about statistical robustness. Commonly used methods or commonly used NMF methods, they assume a particular stochastic mechanism for how mutations are generated by mutational processes. since this mechanism is really just approximation to the reality, and the number of processes is usually unknown, results may sometimes be misleading and or reproducible, as a lot of us have experienced. There's a famous quote by a famous statistician, George Box: "Since all models are wrong, the scientists must be alert to what is importantly wrong." And it's, I think, a goal to make of ours to make models more robust when the model isn't quite perfect to the data itself. And I also agree with Terry and Peter and others that more systematic benchmarking is needed to really understand what models work best when in different scenarios. And there may not be one model that rules them all. One note on robustness that my colleagues are working on that I, again, just wanted to highlight. So Jonathan Huggins, and then several people at Harvard Medical School, Catherine Hsu, Scott Carter, and Jeff Miller. They are working on, something called a power posterior, which can be added on top of an existing likelihood. And what this helps to do is remove some of the noise that may come from model misspecification. And so they've or come from minor perturbations in your data. And so their poster, they showed an example of a simulated data set where they have they generated, they ran NMF using SIG profiler on a data set, a simulated data set without any perturbations, or they perturbed the data set in several ways, including messing with the signatures a little bit, or adding noise counts, or using different distributions. And sometimes NMF would not miss some signatures, or sometimes it would introduce some false spurious duplicate signatures. However, when they ran their tool-based power sig with the power posterior, it would remove a lot of the spurious signatures and still generally estimate more of the correct signatures. So then, as I just finish up my talk here, I just want to highlight some software that my group has recently published, led by a graduate student, Aaron Chevalier, in my lab. It's called Mutational Signature Comprehensive Analysis Toolkit, or MusicaTK. And it's an R bioconductor package. And one of the things we added to it was a graphical user interface. So if you're not a computational biologist and you need a sort of a point and click GUI to be able to run things, run NMF, or visually explore your signatures and be able to click on things or look up more information, you can do that through this graphical user interface. And then let's see, I think, sorry, 1 second, chat came up here. All right, there we go. The other thing this toolkit has is we're adding several methods for discovery, including NMF, LDA, and these Bayesian NMF models that we've been using. So that way you can plug and play and try out different tools, as well as several different prediction methods. And so if anybody has their own tool that they would like to be added, we're happy to incorporate it to try to make more of this sandbox where people can try out different things. And of course, there's a variety of visualizations and plots that people can make. A lot of them have been covered today. We try to give special focus on if you have user specific annotations like tumor type or treatment, to be able to group your signatures or box plots, do various things to really explore that. And let's see. So with that, I think I'm running out of time a little bit, so I'm actually going to skip to the end here and just give a summary. Bayesian modeling can be used to incorporate flanking sequence information or prior signatures into the deconvolution process. The power posterior is one example of something that can be used to make Bayesian models more robust against model misspecification. And MusicaTK is a novel R bioconductor package that provides a sandbox for discovery, prediction, and downstream exploration of musical signatures. With that, I'd like to thank my collaborators, Jonathan Huggins and Masato Yajima, as well as my grad students, Aaron, Shi, Hao Yu, and Kelly. And we have several postdoc positions available for doing this kind of statistical modeling. So please let me know if you're interested.
Excellent. Thank you, Josh. I think we can squeeze in one question here from Teresa. She asks, is it possible that the difference between signatures you observed in different cancers are a result of different preferences for specific genomic regions that might have different nucleotide distributions?
Yeah, that's a fantastic question. And I know others have incorporated genomic region into these types of models too. So that's, I think, another really interesting example where additional information like that can be incorporated into it. So I do think so. I think some of the differences in tumor types could be related to that. And there's certainly no reason why we can't sort of extend these models to include both the flanking bases as well as many of these different sort of regional aspects.
Thank you. So Josh, you can stay on, and the other speakers from the session can come on, and I'll introduce our panel moderator, Bin Zhu, who is a researcher here at NCI within the Intramural Programs Division of Cancer Epidemiology and Genetics.
Thank you. Thanks for the speakers gave the excellent talks. And I prepare a few questions to stimulate the discussions. I'd like to begin my question with the problem puzzle me maybe in many audience for a long time, and also the Peter have mentioned his solution that is like a non-unique solution of non-negative factorizations. So how can we deal with that? Besides Peter's solutions, I just wondering in repair signature or in Bayesian ways, should we still worry about that? And whether the Bayesian way could be address those non-unique solutions issues for non-negative factorizations? Anyone want to try in?
I don't know about Bayesian, but you mentioned Repair6. So this is the method to look at signatures as composition of damage and repair or some primary secondary processes. So in this case, we actually can use as primary processes any definition. So any of these approaches can maybe have different definition of mutational signature. However, our method will use it as an input because we try to use as much knowledge that is already available or and infer only the things that is unknown. So that's another thing that perhaps in many cases, we may have some of the signatures, we may have experimental validation, and that may allow us to narrow down the space of the things that we estimate. And I think that is alternative things, because of course, there's also other approaches that will use lasso, so that minimize the number of signatures. So there may be many different ways we can try to make the solution unique. And I think that there's a very nice argument that by this uniqueness, in the using the minimalizing volume, the correlation between signature 1 and H improves. So that's a very telling. But perhaps another possibility would be just to infer experimentally a bunch of those signatures, and then we'll have some anchor points and less degree of freedoms to infer the less of the mutations.
Yeah, and I would just add there's probably multiple problems that plague people. One is sort of non-uniqueness of solutions or identifiability processes if you're working in the Bayesian space, or even just if you have really complicated sort of landscapes or solutions, just finding the most optimal one can be even challenging. So that's why people just tend to run NMF or do these things many times to just try to make sure they're exploring the space. That is one benefit of taking a Bayesian approach, is that there are a lot of sort of convergence metrics that people can apply to see if at least their model or their different chains or whatever are converging to a solution, even if it's, or a set of solutions, even if they're not unique. But yeah, I agree. It does tend to be a problem. it's a problem sometimes just label switching, which can be easily fixed. But then I think there are other harder problems that Peter was talking about, where you can have actually different solutions with different signatures that still score sort of similarly in whatever method you're using.
I could just add one point about muon volume NMF. There are actually a couple of different flavors of that. There was a paper in August of this year from Shamil Sunyev's group in my department, where they did mutational signature analysis of germline samples from TopMed data. And whereas we do muon volume NMF by regularizing the signature matrix, they do it on the exposure matrix. And that turns out to work beautifully. And so I think depending on whether you're looking at somatic versus a germline, you might use a different flavor of muon volume NMF.
Thanks for all the speakers share their opinions. I just curious for David, in the PICO studies, like in working group, how we deal with those issues of the non-unique solution of NMF.
Yeah, so basically we took the outputs from the signatures working group and we used them within the evolution working group. So we didn't actually deal with those difficult issues within the signature calling. We basically just took the calls as they were, as they came out of the signatures working group. I just kind of comment on the previous question. So I mean, I kind of agree with Teresa that I think using data from experimental models I think could be useful, but I think there's a couple of caveats. I think that the first is that in doing that you're making the assumption that there is only one mutational process occurring within your experimental system, and that may not necessarily be true, because we know there are lots of endogenous processes that will be going on anyway, irrespective of the exposure. I think the second is whether whether we can assume that the signatures will manifest identically in model experimental systems as they do in vivo. It may be that signatures look slightly different in experimental systems anyway. But I do agree that using experimental data maybe within a Bayesian framework, I think could have value.
Okay, so my next question is also kind of related to the first one, also related to the Bayesian method. A couple of solutions have been proposed to address the non-unique solution. Is put additional regularizations or the constraint of solutions. Today we heard about based on volumes. There are also Bayesian method based on sparsities. And likely maybe there are other prior information, Bayesian prior information to further regularize the solutions. So my next question is, when we're facing so many possible choice of the regularizations, which one should we choose, and how can we evaluate which one is the proper one to use in a particular study?
Yeah, I think your second half of that question is really, really important. How do you evaluate? And I think that's something, we've all sort of shown kind of simulations, but there are different ways you can simulate data. There are different distributions you can do there. I think coming up with more sort of rigorous benchmarking methods or benchmarking data sets out there, maybe using a variety of simulation methods, so that way we can see what regularization method works well in certain scenarios, but maybe doesn't in others, or do you have a lot of counts or small number of counts, or those kind of things I think would be super useful. And it's probably missing because I think everybody kind of just does it their own way. And of course, if you simulate the data a certain way that matches with your model, then of course your model is going to perform better. And so thinking about all the different ways we could come up with these sort of benchmark data sets that are as possible, I think would be a nice goal.
One difficulty for me is that there are essentially kind of two ways of carrying out the regularization or sparsity, the imposition of sparsity. One is to reduce the number of signatures that are called within individual samples, and the other is to reduce the complexity of the signatures. So you reduce the number of components that are included in the signatures, and they're essentially working in opposite directions. So you can call a smaller number of signatures within samples by having very complex signatures, or you can call more signatures but have simpler signatures. They are essentially kind of competing with each other, and I suppose we don't know which one of those is more correct.
But obviously, always we can. What I think I always recommend to do is, no matter what sort of the composition we have, we also observe this that non-uniqueness, and one way to one important fact is to be aware what is unique, what's not. Like apobex signatures, they're usually very fine, right? So I think like various classical statistical analysis, like bootstrap analysis, can put some light towards what's being stable, what's not, and what are the boundary of this non-stability for this given set of data. Yeah.
Yeah, I just maybe a more technical point, but we do need to have a way to regularize these solutions. But some signatures are, the correct signature is fairly flat. I mean, it has some features, but we cannot, force sparsity on some of these signatures. And so, we do need to be very careful about these steps. Maybe the theme that I've heard this morning also is, how do we test all these different algorithms that people are coming up with? We've seen this over and over in different areas. Somatic point mutation calling, right? How many algorithms are there? How do we know? And I think it took a while for the community to come up with a really good data set. And there were like the Dream Challenge sort of things. With mutational signatures, I think it is getting a little complicated, partly because the catalog is being updated. And so when you read an older paper, you don't quite know what they mean by this particular signature. That catalog is changing. And so we need a better nomenclature to keep track of these. And then, we do have a great data set in that we have the PCOG, as David mentioned, the signature working group that has worked hard to come up with the latest set. And maybe we need to think about organizing something where we try all these different algorithms on the same data set relatively early on. I say relatively because I think that there will be even more algorithms in the future. And so I think from a user's perspective, there'll be some confusion. I mean, how do you test all these? What's the right data set? How do you know which one is correct? And so I think some collective effort to benchmark these relatively early, I think would save people a lot of time.
Yeah, Peter, you make a good point. I also prepared to raise this question. Is more practical question for the users facing so many choice. How can we choose different tools and by which criteria? So you comments. Maybe any other speaker have their comments how to pick the tools, or how can we use a bunch of tools? What criteria we should use? Any comments on that as well? I think it's very valuable for the audience. No further comments? Okay. I can go back to the regularization. So it just occurred to me, although we can impose lots of regularization, to make those message delivered to the clinicians, to the audience, broader audience, maybe it's better we can make some like biological connections about the regularizations. I'm just curious, for example, like volume-based regularizations, is that possible? That biological connections, like physics, they're not only mathematical tools, they always connect to the real world phenomenon. So can we make such similar connections, not only just for a mathematical property for uniqueness, but they also maybe have some biological interpretations?
Well, if I can start, I mean, certainly within Peacock and other efforts, we can't quite judge or assign meaning to the regularization steps biologically, but we know when we get the signature catalog for specific tumors, what we expect to see and what we don't expect to see. And so that is actually being incorporated into the algorithmic section right now, right? And so when you do this refitting and you see some signatures that you don't think should be there, then you manually sort of find a way to exclude that. And so the biological interpretation is going into some of the algorithms. So that's actually one of the problems, because when you see a paper and you try to reproduce it, you can't quite do it because that step is not reproducible, right? And depending on who does it, you're going to get different results. And so I think we need some way to push that biological interpretation downstream so the method itself is more reproducible. And yet the insights that our biologist colleagues have are tremendously important for figuring out what works and what doesn't. And so we need to perhaps, systematically to catalog what we expect to see for what biological reasons to help us guide in our selection of different algorithms. I'd love to hear other people's thoughts.
Yeah, I mean, one thing the PCOG group did really well, I think, was in their paper outlining some downstream steps you can do to try to evaluate whether or not some signatures that you're getting from these methods, especially if you're doing de novo deconvolution with NMF, like whether or not they are likely to be real. And so, is it found uniquely in a given sample, and do those sample counts really match that signature, or multiple samples, or a lot of these different criteria. I agree, it's probably hard to assign that to specific steps, or those biological criteria, assign them to specific steps in the regularization process, but they are pretty good at least helping to identify some of these spurious signatures. And so that when we talk about sort of evaluation, there's kind of the methodological validation and benchmarking, but there is that sort of biological validation, which I think is useful. So if people haven't seen those steps or those criteria that they outlined before, I definitely highly recommend it, because it's useful to apply it to your own data.
I guess the kind of direct biological interpretation is that if you have a relatively kind of sparse, simple signature, that suggests that the mutational process is very specific and it's operating on a very specific motif. Whereas if you have a less sparse signature, like signature 5, that suggests that it's much less specific. I guess the difficulty is, as Peter mentioned earlier, that if you're imposing certain regularization on all of the signatures, then that might do well for detecting the more specific sparse signatures, but then it might rule out the less specific signatures. And we know actually there are lots of different mutational processes, some of which are do operate in very specific contexts, but others that are much less specific.
Yeah. Okay. So given time limit, maybe I can only ask the last question. It's more like an open question. Is so in this section, we know there is a two kinds of analysis. One is discovery the de novo signatures, another one is given the signature profiles. We estimated the activities, study its clonal evolutions, etc. Besides work you have been delivered or talked, what other unsolved problems or opportunities in each of those areas you think the field should be emphasis on and we should work together?
I know one challenge that my group is interested in, and we had a poster on it as well, is thinking about the joint sort of SBS signature estimation and other things like Indel or DBS. There's problems there where if you combine those two matrices together and try to do one NMF deconvolution, then like sometimes the SBS dominates because they're so much higher and you miss subtle patterns that happen in the lower express counts. So then a lot of people do it separately, like they did in Cosmic, and then they try to correlate them post hoc across, to try to say, oh, hey, this is involved in similar things. And so trying to figure out better methods to sort of jointly estimate signatures across multiple sort of mutation modalities at the same time, I think is another interesting problem.
Actually, the major time sink for us is not developing an algorithm, but actually trying to get access to a data set, right? I mean, we want to collaborate with the clinician wants to collaborate with us. The moment we try to get raw data to us, we have to sign RFPs, this committee, that committee, lawyers get involved on material data transfer agreement. And I think there will be a lot of methodologists interested in testing their algorithm, except that they won't be able to get their hands on the data. And this will be a major problem. And the data we do get access to will have missing, clinical phenotype data and such. And so, the Project Genie that I mentioned, I think there is a promise there, but I think some sort of curated data that a lot of people could use to test the algorithm is going to be really important. Otherwise, we're going to have these piecemeal efforts with small n's, and it's going to be hard to figure out what works and what doesn't.
Yeah, good point. I think that's like in GWAS areas, people come to feel the same pain and they come to a solution. They just release high-level summary data and free for all. So the field indeed speed up a lot, take advantage of those. I think maybe our field should be follow the same steps in future. Any other final comments on the future directions for methodology work?
I think there's still work to be done with other types of aberrations. So I think most of the talks today are focused on the SBS signatures, and I know there have recently been published some like copy-number signatures, for example, from Jeff McIntyre. But my feeling is that though copy-number signatures are slightly less mature than the SBS signatures, I think there's still some work that could be done on the copy-number signatures. We're quite interested in those in my group.
Okay, if there are no further comments, because the time is up, I will conclude this discuss section. Thank you everyone for joining and make comments and discussion.
Yeah.
Thanks.
Thank you.
So I guess we are getting to the closure of this first day. It's been really, really interesting, and we think that we have at least addressed some of the issues that we raised at the beginning. Behind the scenes, Ludmila and I were trying to get the major points that we wanted to highlight after this closing of the day before we reconnect tomorrow. So maybe I'll share the slide with some of these comments, or do you want to do it? Okay.
I just shared the stories there, yeah.
Okay. Okay, Ludmila, why don't you go?
Sure. So as Terry said, it has been a very vibrant day with a lot of discussion and a lot of points coming through. We have tried to distill them into 4 general high-level points. One is the reproducibility issues. We have the different tools. We have different sequencing platforms. We also, for different platforms, we have different coverage, or samples tumor samples can have different purity and ploidy, which further complicates it. There are different classifications. Just for single base substitutions, you can get at least 3 or 4 different classifications. And there was a very clear desire for larger unbiased data sets for benchmarking mutational signatures, as well as for updating the nomenclature to reflect changes into the signatures catalogs. A number of common questions emerged throughout the talk. Again, the distinguishing of flat signatures is a problem. We saw some beautiful method coming from Peter's group that's hopefully going to address some of that. We saw some questions of what happens if you have 2 different mutational processes generating the same signature. For example, SBS4 being generated from tobacco smoking, or from non-tobacco related PAHs, or from something else. We talked about approaches to identify the causes and etiologies of mutational signatures. We briefly touched, and I think we'll talk about more tomorrow, about when is signature clinically actionable. Is it enough to detect signature 3? Should one go to the level of companion diagnostics to be clinically actionable? We discussed a bit tissue specificity. And again, we talked about normal tissues and non-tumor diseases, both about signatures and driver genes. Also, there was a beautiful overall discussion about timing of mutational signatures and evolution. There were differences between the tools for timing and reconstruction and evolution, and potentially the additional information that mutational signatures can add. We talked about also using simulations and experimental data to validate some of these predictions. And one additional general comment was the accessibility of mutational signatures, accessibility of data sets. These platforms are way too expensive for most parts of the world. I mean, some of us are in the United States, in the United Kingdom, in the Western part of the world, but for most other places, they're not usable. So that question has been posed, and I don't think at this moment we have a good answer. So these are our general discussion points that we have curated with Terry throughout the day. We're going to curate the same sets. Well, we're going to curate discussion points tomorrow, and then in the end of this workshop, we're going to have about 30-something minutes to discuss them. With that, I want to thank everyone for attending. And I think we start bright and early tomorrow, or at least for us in the West Coast, 7 a.m. West Coast, 10 a.m. Eastern time, with Ned Sharpless presentation. Dimitri, sorry, I can see you want to say something.
Yeah, just very quick question. Could you explain what is accessibility of signature? What do you mean? And is it just accessibility to the list of mutation calls that were used by others? Because that is a cheap stuff. And this is what is missing really for people who redo analysis. And Peter just alluded to the same in his discussion.
So I meant to an extent that getting the actual data, which can take a lot of time and a lot of effort, and the exponential rule of lawyers, getting more lawyers to agree about IP. But I also mean the actual ability to detect signatures from other parts around the world. If you're doing cancer biology in Mexico, Iran, Russia, etc., the places that don't sequence whole genomes, or the places that don't generally sequence, using mutational signatures for epidemiology or clinical applications is just not possible. It's just not cost effective. So thinking of something that people in places that are not as affluent can use to utilize mutational signatures.
And if I can add also following Peter's final comment, that in addition to the data with the mutation signatures, we really needed to link this data with clinical data or epidemiological data or exposures and so on. Otherwise, we limited the possibility of using these signatures in other settings.
Mm-hmm. I will address in panel of tomorrow's discussion panel, specific questions about mutation calls and accessibility of mutation calls. But I think it is essential for developing a comparison panel for different signatures using the same large sets of mutation calls.
Right.
which you actually indicated as very important thing.
Yes. Okay. Well,
Thanks again,
thank you,
everyone.
everybody. Yes.
See you tomorrow.
See you tomorrow. Bye-bye.